Hello, my name is Marko Hyvinen, and I'm telling you here a brief story of a protein engineering project we have done in my group at the University of Cambridge. This is our attempt to develop a modified version of Activin A, which is resistant to inhibition by follistatin. The work was done mainly by Chulu Wang, with the start of the project done by Kat Donaldson in her PhD. Now let's start with Activin A. Activin A is a TGF-beta family growth factor, which you see here. It is a dimeric structure like all TGF-beta family growth factors, a very heavily disulfide-linked dimer, which signals by binding to two different receptor types, one of them type 1 binding here on the wrist epitope and the type 2 binding on these knuckle epitopes here. In adult organisms, you can find it in many tissues. It is generally involved in early repair and inflammation processes, for example, in wound healing. But it found also a lot of use in stem cell research because activin, being a surrogate for nodal, can mediate maintenance of pluripotency in stem cells. And when you up the concentration of activin A in the media, the stem cells will turn into endodermal lineage. Now, when you look at the signaling cascade of Activin A, here shown schematically, the green cartoons are the two different receptors and an activin dimer here in yellowish brown. Binds to the receptor, and binding on receptor induces intracellular phosphorylation of SMAD proteins, which translocate to nucleus and activate transcription. One of the target proteins, among many others, is follistatin. Follistatin is secreted protein. It goes outside the cell and it binds specifically to Activin A. And by doing so, as you can see on this diagram here, purple follistatin engulfs activin dimer and stops it from binding to the receptor. So this is a typical feedback inhibition loop that we see in biology a lot. Now, we knew that this was happening and we were wondering, well, if we culture stem cells in activin regularly, does the follistatin accumulate in these cells as well? So what Chulu did was that she took induced pluripotent stem cells and cultured them in the typical activin-containing media. She did that for 7 days and took daily media samples from the cells and first analyzed those by ELISA to look at how much activin there is and how much follistatin there is in the media. And you can see in this graph here, in those yellowish brown bars, you can see activin stays relatively constant in total concentration, but very soon follistatin accumulates to very high concentrations in there. And you can see this is a molar concentration. So on day 2, there's already more than twofold excess of follistatin compared to Activin A. Now we measured Activin A as a total concentration because the way the ELISA works. So the question is there bioactivity left in there? So we took those same samples and put them through a luciferase-based bioassay using HEC293 cells, an assay that responds to activin in the sample. And you can see that while we have 100% activity at the start of the experiment, on day 2 already there's pretty much no activity left in the medium. And that was attributed to the increase in follistatin concentration because we know that inhibits activin very efficiently. So what if we can then make an engineered form of Activin A, shown with this little bit, distorted now molecule here, in such a way that it can still bind to receptor and induce the signaling? It would make follistatin, because that would be difficult to stop, but the follistatin couldn't bind as efficiently anymore to activin, so it wouldn't engulf all the activin in its stronghold. We are structural biologists, so we took inspiration from structures. First of all, you can see here how this long molecule, purple and pink follistatin, 2 of them, bind on this central Activin A, blocking both type 1 and type 2 receptor binding sites. just like in that cartoon, it really surrounds activin completely. It's the structure of full follistatin 288 with Activin A from Tom Thompson, but our inspiration came particularly from the structure in our lab by Adrian Harrington, where an FS12 domain fragment of follistatin in complex with Activin A, we can see this arginine 192 inserting its little positive finger in between the fingers of Activin A. So we look at these structures, generated mutants, and I'm giving you an example of data from a couple of them. So you can see, first of all, we could detect reduction in binding of follistatin to these engineered activins. So these purple and gray lines here show the binding in bilayer interferometry or octet biosensor compared to the wild-type Activin A. It's clearly reduction in binding. We put those same proteins in inhibition bioassay, so we have constant concentration of Activin A of different forms, and we titrate in follistatin in increasing concentration. So you can see the follistatin-resistant activins, the purple and green, are shifted right compared to the wild-type Activin A. So we have reduced affinity and we have reduced inhibition in bioassay. And importantly, the primary bioactivity of these mutants is retained. And this was not the necessarily obvious thing to happen, as the binding site for follistatin and Activin A overlaps perfectly with the receptor binding sites. So we had to be very careful of choosing these mutations so as not to affect the bioactivity. So with these two improved activins in hand, we put them in the same induced pluripotent stem cell assay. You saw this data already before. So the purple bars there are the follistatin increase in the cell culture. And now when we treat the cells with the follistatin-resistant Activin A, we get still follistatin accumulation, perhaps even more than with the normal Activin A. But when we then analyze those same samples in this luciferase bioactivity assay, you can see the bioactivity even after 7 days is still over 40%, while in the normal Activin A it was pretty much zero after 2 days. So we seem to, looks like we've achieved inhibition by follistatin. We tested the effect also in human embryonic stem cells and these SHEP4 and SHEP6 cells here. You can see in yellow the normal Activin A bioactivity goes down rapidly in culture, while the follistatin-resistant Activin A retains over this 6-day period, more or less full bioactivity. We also, Shulu did the similar normal kind of a 2-day media change culturing of the cells, and you can see here in these examples, we have this constant up and down yo-yo effect of bioactivity with the normal Activin A, whereas the green follistatin-resistant Activin A retains more constant bioactivity through the culturing. So just a summary, this follistatin-resistant Activin A is a variant where we have shielded the protein from follistatin's inhibition while protecting the bioactivity. And the work now progresses to see if there's any particular application for these engineered proteins. And we hope to get the paper out soon for you all to interrogate the finer details of this work.